This lecture is just going to be a really quick introduction to field of biochemistry, which I kind of think of as kind of the chemistry of life processes. Lots of you guys that are ending up in A&P. A lot of what you're talking about having to do with chemistry is going to be directly applied to this field. So I thought I'd give you just kind of like a chemist, real quick bent on exactly how you can understand these things. There's a couple of concepts you need to know before we launch into our mini discussion here. And that is one, that there exists this thing called polymers. A lot of times people will call these things macromolecules. And all a polymer is, right? You might read poly and recognize that there's many. So it's read many mers, and so the mer is a monomer. So really, polymers or polymers are just compounds made of some sort of repeating monomer, some sort of small molecule unit. And it turns out that the types of macromolecules that we see in living systems, which, by the way, are sometimes called bioorganic polymers, if you want to impress grandma with that word. There's really four major classes, and a lot of these you might have some experience with if you know anything about nutrition and things like that. So we're going to be talking about proteins to start with. And keep in mind, for each one of these four groups that we're going to see in just a second, I'm going to kind of give you just some real quick tips in terms of how you can recognize each group, what monomers make them up, and we're really going to be limited on talking about functions in living systems, and that's really what A&P is going to be all about. So we've got proteins, we've got carbohydrates. You probably have heard of carbs before. Lipids, which are also known as fats, and then also nucleic acids, which are made up of both RNA and DNA. So those are the four groups we're going to talk about. It's important that you recognize that we do have a bunch of different functional groups, and we're going to highlight a couple of these as we go through these four macromolecules. One of the things that is true of macromolecules, they're just big, giant, hairy organic molecules. Keep in mind that organic means that you're made up of carbons and hydrogens, these big, giant chains of carbon. It turns out when you put things other than carbon, or kind of things that are a little more interesting than just what's called an alkyl group, which is just like a single bonded carbon with its hydrogens, that's called a functional group. So all of these different interesting things we kind of talked a little bit about in one video lecture, where we talked about maybe a carboxylic acid and an amine coming together to form an amide, or a carboxylic acid and an alcohol coming together to form an ester. It turns out that those types of reactions, the reason I highlighted them when I could have highlighted any bazillion reactions, is that those are really important reactions in terms of how we kind of put our macromolecules together in those four different classifications. We'll start off talking about the monomers for proteins, and these things are called amino acids, which I'm sure you've heard of before. They really do have kind of four different parts. One is the carboxylic acid. You'll also notice that this little carboxylic acid, normally we're used to having an H right here. It's actually lost its H, which means that it is acted as an acid. So what we're looking at is actually what's called a carboxylate. It doesn't really matter. You can think of it as the H attached out there. So on an amino acid, it's pretty easy to tell that you're an amino acid because you'll have some sort of carboxylic acid group on one side. That's the acid of the amino acid name. And then you have this other thing, which is, in this case, it's protonated. So in other words, this is kind of acted as a base. So you can kind of imagine that H isn't there. And what we have in that case would then be called an amine group. And so that's the amino of an amino acid. It actually tells you what's in there if you know your functional groups. So an amino acid. And then the other part of a monomer, which is called an amino acid, is this R group. And the R group is a way of representing like a whole bunch of different groups. It turns out there's 20 of them in, or at least naturally 20 different amino acids. And the way that they're different is this little R group that can change it up. And so depending on what you want your protein to be doing, you put different R groups in there. And of course, all of those R groups have different functionalities or functional groups that we're really not going to talk about. And you really probably wouldn't talk about that even in an A&P class. That would be more of like an introduction to biochem class. So here's kind of one way of thinking about a polymerization process. In this case, you'll notice that it's not necessarily just an amino acid polymer, sorry, a protein polymer, but instead we're just kind of showing this idea that these little tiny units that are in this case represented like beads. So since we're talking about amino acids, it might be a good idea to imagine that this thing is an amino acid monomer. And pretty much all of these polymers are going to come together by the liberation of water. And then you'll notice that when water kind of squirts out the other side of this thing, what happens is that you increase the length of that polymer chain by 1. So, and this is something that I made a little video for you to kind of show you specifically the case of amino acids and also kind of show you where water kind of comes from. So stay tuned for that. So for this quick little video, I wanted to show you all what it looks like when two amino acids come together. This is an important reaction to know about because this dehydration synthesis, as it's called, that kind of joins two parts together, is super important in A&P and all the different macromolecules that we're going to talk about. So I just decided to show it to you right now with amino acids and some molecular models. So first of all, let's take a look at this. And just recognize that it is in fact an amino acid. Here you see the carboxylic acid group. There's the carbon, there's the carbonyl oxygen, and there's that little OH group that's hanging off the side. Obviously the reds are oxygens and the blacks are carbons. There's that center carbon of the amino acid, and then this guy, this green one, that's just representing what's called that variable group, and that's where you get your 20 different amino acids depending on what's connected there. We're just kind of doing it in general now. That's a green. There's the H that's generally attached to amino acids, that center carbon. And there's the what's called the amino terminus or the amine group kind of on the end here, right? And obviously blue is nitrogen. So how the heck do these two things come together and how does water kind of come out of it? Well, it turns out if you kind of think of a carboxylic acid as sticky, right? Imagine that this carbon right here is sticky and really wants to kind of bond with things. It turns out that on our other amino acid over here, that this nitrogen is also sticky. So what's going to happen is that blue sphere and that carbon are going to kind of come close to each other. And as a result of that, a water is going to pop off. So where does water come from? Well, before carbon can bond, right? Carbon can only bond 4 times. So this little OH group that's kind of hanging off the side, we're going to take that off, right? And what you'll see there is that that is going to allow a new bond to occur there. And so we're going to take exactly 1 hydrogen off of our nitrogen here. So here's the hydrogen I'm going to take off. I'll set that aside for a second. That nitrogen is going to covalently reach in and create a new bond. And you'll notice when I do that, suddenly I've taken those 2 monomers and I've turned them into a dimer. And I've made an entirely new functional group here. This functional group right here, where we've got a carbonyl oxygen attached to a nitrogen on one side and a carbon on the other, that's called an amide bond. Or if you're a protein chemist or an A&P, this is what they're going to talk about as the peptide bond. And that's actually what joins proteins together, or I should say the amino acids of a protein together. And if you look at what I kind of had to take off as a result of that dehydration synthesis, it would make sense that it was a water. And so that part that I took off of the carboxylic acid right there, and the H that I took off of the amine, if I now connect that together, you'll notice that we've got good old Mickey Mouse here, which is a water molecule. So as a result of my reacting a carboxylic acid and an amine, I ended up with a brand new bond here called an amide. And what I could do is I could keep doing that on that carboxy terminus right there, and I could keep growing this thing. And that's exactly what a protein is. This kind of reaction, this what's called dehydration synthesis, where a water pops out, is really common. This is kind of the way that we build things in our body. And then if we need to break something down, say like I just ate a steak and I want to break down the protein, what happens is the exact opposite of that. That water molecule comes in and cuts apart this bond, and everything kind of goes back to normal. Right? So here's my water. I put that part of water on the carboxylic acid, and then on this other end, we put the hydrogen back. And then we end up with our two monomeric units once again, and this is a reaction called a hydrolysis, right? We add water, break things apart, we remove water, we join things together. So hopefully that'll make a little bit more sense than some of the PowerPoint slides. Hopefully that video was pretty helpful. So what we see here are a couple of kind of molecular structures that include a couple of different variable groups here. This guy's called methionine, and then you've got another thing called aspartic acid, and you can read. But the idea is that these four different amino acids, if they have those sticky ends, undergo some sort of dehydration synthesis. What happens is what's called a polymer change, or a polymer chain, or in this case is called a peptide. A peptide is kind of like a miniature protein. But each one of these groups that kind of sticks off the side here imparts different types of interactions in chemistry to a particular protein. And so one thing that you'd probably talk about a little bit more in like a true biology class or something is, the fact that these protein polymers end up being these kind of ribbon-like structures that kind of look like a big giant booger to me, and you'd actually talk about, what exactly is going on to kind of maintain the structure of these guys. Turns out the structure of a protein is super important, because without that structure, it's not going to work properly. So a lot of diseases, for instance, are caused by misfolded proteins, or proteins that are missing certain amino acids, or there's so many things. Like we could literally talk about this for 4 years and still only just skim the surface. So we're not going to do that. So there's all kinds of interesting structures here. As I said, we're going to talk a little bit about what proteins do just really quickly. Probably the most important thing that they do is they act as catalysts in the body. And what a catalyst does is it allows chemical processes to happen that would otherwise take a really, really long time, and speeds them up and lets us kind of be the amazing creatures that we are. Another thing that proteins do is hormone response. So anytime your body receives some sort of signal, it's often from some sort of protein, which we call a hormone. And of course, there's a ton of other things. There's like, structural proteins like collagen in your knee, and just so many different things that a protein can do. Muscles allow us to move around, and those are little proteins called actins and myosins and things like that. You'll talk about a whole bunch of that in A&P. The next type of macromolecule that we're introducing is carbohydrates. You're probably aware that one of the most important jobs of a carbohydrate is metabolism, or we use them for energy. There's also a bunch of different things that they do, but that's probably the number one job that you'll talk about carbohydrates for. Turns out that the monomer for a carbohydrate is always called a monosaccharide. This is just one of many different monosaccharides, but it happens to be a pretty important one, and that is glucose, which you're probably aware of. This guy can exist in a linear or a ring form, so in terms of you guys like recognizing it on an exam or something, pretty much anything that just has a whole bunch of alcohol groups attached to it, and oftentimes also an aldehyde or a ketone out there, that's usually some sort of carbohydrate. And so the idea is that when you build these things into polymers, if you take a couple of glucoses and connect them together, it turns out that you form something called maltose. And then you can also take different monosaccharides and connect them to glucose. And so you've probably heard about sucrose or table sugar. This is, in fact, made up of something called fructose and glucose. So two different monosaccharides coming together. That's called a disaccharide. There's other things like lactose, which is milk sugar, which is another type of disaccharide. But keep in mind, these things are all made of repeating units of those simple sugars. And then, of course, you can get these big, giant carbohydrate networks. This is cellulose, which is a component of like bark and leaves and things like that. It's a kind of the thing that gives plants their structure. And it itself is made up of a whole bunch of glucose molecules. If we were to look at starch, there's is also a polymer of a whole bunch of glucoses connected together. That's a really good energy source. But that's just pretty much all I want to say about that. Just be aware of the fact that these things have a whole bunch of different alcohols connected to the little monomeric unit, which is called a monosaccaride. The third bioorganic polymer that you should at least be introduced to are lipids. These things are the what's called fats, probably more commonly. This is a pretty interesting one because it's actually made up of several different monomers. And there's also kind of variations on the theme, but probably the most important one that you'll talk about is made up of both glycerol and these things called fatty acids. One of the things you'll notice is that glycerol's got these alcohol groups that kind of hang off of it, and there's 3 of them. And then each fatty acid has a carboxylic acid. So just like we saw before with proteins, when you bring these two things together, if you remove a water, you can actually have that oxygen sticky end attached to that carbon sticky end. And that's how you build up something called a lipid. So here's just kind of an example of one kind of lipid. This is actually going to make what's called a complex lipid. So you can see as a result of taking a water out of this guy, we form this new bond. And then taking a water out of this guy, form this new bond. And then, of course, this is just a big crazy thing that is super duper polar. And the reason that this is kind of something I talk about is that it turns out that this forms the cell membrane layer. So we're all made of cells, and then the little envelope that's around our cells is this like super greasy thing. Lipids are just basically things that contain a whole bunch of different carbons and hydrogens without those really polar oxygens present on. But those really polar oxygens are present on one end, which kind of makes this part kind of like water a lot. So that'll kind of be facing towards the water compartment of our cells. And then this big long chain right here, the greasy part, is kind of facing towards the inside. And what happens to make that little envelope is you basically have those really greasy things on the middle here away from the water. And then you have the more charged things kind of towards the outside. So we call that hydrophilic and hydrophobic. So the greasy tails here, they hate water, hydrophobic. And then these guys down here and also up here facing the outside of the cell, those things are called hydrophilic. And they like to kind of be pointed towards water. Here's a little more interesting look at what's actually in the cell membrane. You'll notice that these are all those little phospholipids that we were just talking about. And it turns out that in there are also different proteins that provide channels for different things that you want to get inside of the cell. There's often little pumps that kind of pump things through the cell actively. You've got all kinds of different like carbohydrates hanging out on the outside that gives rise to like things like blood type and all that. They're kind of like little flags that say, hey, I'm a foreign invader kind of thing, or hey, I belong to you. Don't eat me. So these kinds of lipids that's probably the most important function of those guys. Turns out that there are a whole bunch of different types of lipids that are not based on fatty acids and glycerols that you'll talk a lot about in A&P. And the other type of lipid that you'll talk about are these like corticosteroids and such that are all based off of cholesterol. They tend to kind of reinforce that cell membrane, and what I call sexy time hormones like estrogen and testosterone. You'll notice that we get lazy as organic chemists, and we often don't draw the carbons and hydrogens here. So everywhere that you see like a bend in the chain, that's actually a carbon. So these things are super duper greasy. But it turns out that they also have a pretty important hormone response. There's just the similarities between estrogen and testosterone, and of course there's a bazillion other ones. Another one you'll talk a little bit about in A&P, which is a lipid-based kind of signal molecule, is this thing called prostaglandin. Anytime you injure yourself, your body creates a whole bunch of this stuff, which kind of causes that red inflammation that is kind of a way for the body to recruit the repair mechanism. And we're not going to talk too much about this, but you certainly will talk about it in A&P. You'll probably talk about the cascade that results in the production of this when you injure yourself. And just interestingly, this is one of the things that things like aspirin and ibuprofen and such kind of interfere with the production of this lipid. The fourth macromolecule is probably the most interesting and most complex structure. You probably heard about nucleic acids and the fact that they serve as the genetic material. You'll definitely talk about this a whole bunch more. But this guy is pretty easy to recognize because you have this phosphate group, which really isn't a part of too many other, major macromolecules. And then also this 5-carbon sugar. In this case, that one's actually a deoxyribose sugar. There's also a ribose sugar, and they're both used for two really important different jobs in the body. The deoxyribose is more the genetic material, the thing that actually stores all the way that we make all our proteins and really make us. And so what we have here is this kind of what's called a backbone. And then there are these other things called nitrogen-containing bases that actually form these nucleotides. And anybody that's ever taken a biology class has probably talked about the fact that adenine always pairs with thymine and cysteine pairs with guanine. So that's kind of these 4 different groups, at least in the case of DNA, is what you put in here, depending on what protein you're trying to code for. Another thing which you'll want to be just really quickly introduced to is the D of DNA, this deoxyribose, and the difference between that and ribose. Of course, these two sugars are involved in very different nucleic acids, right? We've got DNA and RNA, but the real only difference is that you put this alcohol group in there, and it turns out that this thing, this is kind of more of a messenger, kind of like a worker bee, relative to this thing, which actually, with the phosphate backbone and the nucleic acids, actually stores the genetic information. So you'll talk a lot about how these guys grow in future classes, and but it's important to recognize that there is a difference between DNA and RNA that has to do with that little sugar. So there's probably the view of DNA that you're very used to. That double helix is, pretty much one of the most important developments in science of like all of the 20th century. And so what you really have is kind of what I think about as a twisted ladder, right? This is that double helix. And so this little ribbon right here, that's that alternating phosphate and deoxyribose thing. And then that little nitrogen-containing base area that we just saw, those are kind of the rungs of the ladder. And you'll notice here that a T pairs with an A on the other side. And that's something you'll talk a whole bunch more about. And it turns out that it really has everything to do with chemistry, right? This idea that A bonds with T. Why? Well, because it turns out that their molecular structures are such that they perfectly hydrogen bond together. G's bond with C's for exactly the same reason. So there's 3 little perfectly situated hydrogen bonds between these guys. So what you'll typically do in an A&P class is you'll talk a whole bunch about how these kinds of base pairings actually store genetic information. So you have that to look forward to.